We're recording already. Hello, everyone. I'm Johannes Weikmeier, Assistant Professor at Stevens Institute of Technology. I'm hosting today's A Closer Look Journal Club, co-hosted by Emma Lejeune, Manuel Rausch, and Adrian Buganza-Tipole. We have the honor of listening to the work of Matthew Bersi, who is now an assistant professor at the University of Washington, St. Louis since August 2020. He did a postdoc at Vanderbilt before that and got his PhD from Yale. He recently was awarded a K99 NIH award, which is an incredibly big recognition of someone who's going to do some prominent and great work. So what he will be talking about today is on the topic of mitral valve degeneration due to regurgitation or the backflow of blood after a valve, and the idea that ablation might be useful to alter geometry, microstructure, and mechanical properties. So Matt, the floor is yours.
Great, thanks for the introduction. That was very nice, and I'm happy to be here and to present some of this work. As Johannes said, I've recently started my own lab at WashU St. Louis, but this work that I'm going to be talking about today was work that I did as a postdoc at Vanderbilt University in the lab of David Merriman. And this is kind of builds on a lot of work that had been done historically in his lab. So I'll give some context for why we're studying this and some of the previous work, and then some of my contributions. I should also say that this work was primarily done by an undergraduate student at the time. His name is Josh Bender. He's since moved on to medical school at Columbia. And Wilson Adams was still currently a PhD student at Vanderbilt in the biophotonics group. So with that, I'll just go ahead and jump in. Okay, so as Johannes said, today I'm going to be talking about mitral valve prolapse. And in particular, when we talk about the mitral valve, this is the valve that separates the left atrium from the left ventricle on sort of your systemic side of the heart. And under normal conditions, a valve microstructure may look something like this, where we have this leaflet sort of in cross-section that's lined by endothelial cells. And inside the matrix of the valve itself, we have populations of quiescent valvular interstitial cells, quiescent meaning they're sort of dormant. They're just sitting there, feeling their environment. They're not really synthetic or activated cells. And these cells reside in a matrix of primarily collagen, but there's also elastin and another specialized protein known as glycosaminoglycans. As we age naturally and under certain genetic conditions and under cases of altered mechanical loading, maybe through diet-induced changes to the ventricle, for example, this otherwise healthy extracellular matrix can become diseased. And that may look something like this, where we have certain hallmarks that I'll denote here. So the first of which is what's known as a myofibroblast activation. So these cells that were initially quiescent and sort of residing in their matrix, they become activated. And when I say activated, this means that these cells begin to produce enzymes and cytokines that actually alter the actual structure of the matrix that they're residing in. So one of these is known as matrix metalloproteinase, or MMP. And depending on the type of MMP, this could degrade collagen or this could degrade elastin. And ultimately what this results in this diseased extracellular matrix is remnant fragments of elastin and collagen throughout the interstitium of the valve. And there's also, in particular in mitral valve prolapse, there's this hallmark of an excessive glycosaminoglycan production. So the leaflets themselves become thicker and sort of a swollen phenotype. And what that leads to is a sort of quote unquote floppy valve. And that's actually a clinical term, a floppy valve. And if we see floppy valves sort of hemodynamically, maybe by a sort of routine clinical ultrasound, they may look something like this. This, for me, in the untrained eye, is a little bit difficult to see what's actually going on. So thankfully, this reference has provided some schematics here. But what we can see is that for these degenerated floppy valves, we have an impaired coaptation, where coaptation is a word that describes sort of the normal closure of the valve leaflets. So if you think about in a normal situation, with each beat of the heart, the valve should properly close to prevent backflow from the ventricle into the atrium. Of course, whenever we have this altered coaptation, that's not the case. And we have this presentation of what's known as mitral regurgitation. So this is sort of the major hemodynamic sort of clinical observation associated with mitral valve prolapse. And here again, we've sort of annotated the left ventricle on an ultrasound in the left atrium, the mitral valve separating the two. You see that with improper closure, we get this stark jet backflow into the atrium. So this is what people are looking for when they talk about mitral regurgitation. Obviously, you may think that this is not a good thing, and it turns out that mitral regurgitation can lead to several complications. The first of which is known as dyspnea. Typically, this is just a shortness of breath because oxygenated blood is not flowing properly throughout the body. Also, we can have presentations of things like atrial fibrillation. So this is sort of a non-synchronous, non-uniform fluttering of the atrium, can lead to other complications like stroke and so on. And then ultimately, patients who have persistent severe mitral regurgitation can actually have long-term remodeling of the ventricles themselves, so that the heart actually adapts and can lead to altered function, and then we can have ultimately a presentation of heart failure. So this is obviously a problem. Typically, this is occurring in an older patient population that may have other comorbidities. And so treatment is obviously of paramount importance. So it turns out that in terms of treatment strategies, there are really no targeted pharmacological treatment options, which means you can't give a drug by mouth to treat a degenerated valve. This is true for all valves in the heart, mitral valves, aortic valves, tricuspid valves, if you're talking about the right side of the heart. But it turns out that there are multiple surgical approaches that are effective and have evolved over time. So the first of which is what's known as a leaflet resection. Looks something like this. If we have in this schematic a leaflet that has sort of a flail behavior, sort of this floppy valve leaflet, what can happen is a surgeon can go in, actually resect the portion of this leaflet, perform as operation to sort of stitch this closed, and if needed, even provide what's known as an annuloplasty ring to sort of support the actual entire valve apparatus itself. This procedure is very successful, has very high success rates, but the counterpoint to that is that this is an invasive open heart surgical procedure, typically. And since it's invasive and open heart, even though it has high success rates, this can be a difficult procedure for patients to undergo. So again, patients that are typically older, have other comorbidities. So this can just be a difficult procedure to endure. And so because of this, strategies have developed to actually have other approaches, one of which is known as an in-valve replacement. It looks something like this, shown schematically here. And this in-valve replacement nowadays is typically done through what's known as a percutaneous catheter-based procedure. So I think the word percutaneous just means through the skin or to the skin. But the idea here is that you actually introduce the catheter typically around the groin into the femoral vein and sort of snake this catheter up into the right heart circulation. So this is a right atrium. And then you perform a sort of transseptal approach through the atria down into the left side of the heart. And then you can deploy this valve typically just right inside of the degenerated valve. And this valve can be artificial. This could be sort of a prosthetic, or even now people are trying to bioengineer these valve leaflets. The advantage here of the percutaneous approach is that this is minimally invasive. So typically, again, it's just an incision in the leg. But there can be other catastrophic complications associated with this approach. For example, if there's an improper anchoring of this sort of indwelling leaflet, you can have displacement that alters, again, the function, and this can obviously be a problem. Specifically for the mitral valve and sort of mitral valve degeneration, Abbott Medical has developed a percutaneous approach that's known as the MitraClip. And this is not exactly an in-valve replacement in and of itself, but this is truly a clip. And so there's a percutaneous approach where this clip is administered very precisely in certain locations to actually pin these two portions of floppy valves together to actually reduce the orifice area of the valve itself. Again, this is minimally invasive. It requires a very skilled surgeon with years of practice to actually place these properly. But the caveat here, again, is that you have this sort of permanent indwelling medical device, and sort of FDA approval is for 15 years of efficacy, at least up to this point for the MitraClip, but long-term effects may not be fully understood at the moment. So again, I should say that all of these work and are used, probably on a daily basis. But some of the work that I'm going to be talking about today is sort of envisioning a situation where an ideal surgical option would be one that is minimally invasive, so typically a percutaneous type approach, but doesn't leave any sort of permanently indwelling device. And so to this end, what we wanted to do and what was done previously in the Merriman lab was to look at this idea of radiofrequency ablation. So radiofrequency ablation, or RFA, it kind of hits some of these checkboxes. So this is a minimally invasive procedure. You have a radiofrequency probe that's can be snaked up just like a catheter into the heart. And the idea here is that you can actually generate very local, high temperatures that can be applied to the tissue and sort of alter properties of the tissue. And RFA is used clinically in many different applications. One of the main ones is pain management. So typically people will have sort of a chronic nerve pain. They can actually do a nerve block with the RFA probe and sort of ablate that portion that's causing pain. Also for cancers, we can have solid tumor therapies or even varicose vein treatments. But one of the things that we were interested in is the fact that RFA is used in the cardiology field regularly for cases in the sort of electrophysiology. So an example of what this may look like in the heart, this is a sort of cross-section of the myocardium that has been ablated by RFA. What you see here is sections of normal myocardium to the left and right that are sort of sandwiching this region of myocardial necrosis that's due to the actual ablation itself. So this high temperature causes a lesion to form, sort of cell death, and you get this, in this case, through thickness lesion. As I said, RFA is used in electrophysiology pretty regularly, and the idea here is that your RFA probe can actually be used to alter conduction pathways in the heart. And so for patients who have atrial fibrillation, again, which is associated with mitral valve prolapse, you can perform very precise ablations, typically around like sort of the ostium of the pulmonary vein. And this can alter the conduction pathways in the heart and sort of restore normal fibrillation. And so we wanted to potentially take advantage of this because we know that RFA alters tissue properties as a result of this thermal denaturation of the cells and extracellular matrix. The idea here would be that if we could harness this technology, we would be able to alter the properties of the leaflet without having something that's sort of maintained permanently in the patient for the rest of their life. So this brings me to some of the preliminary work that was done in the Merriman lab looking at this. And what they had previously shown is that the ex vivo application of RFA can alter the geometry of mitral valve leaflet. So this is just a schematic of sort of the mitral valve annulus that can be dissected out, and a sort of rectangular or square portion of the anterior leaflet of the mitral valve is excised, and 4 markers are placed on the surface. And then RFA is applied ex vivo to this excised leaflet. And what they found is that with increasing amounts of radiofrequency power, you can actually see a increasing change in the leaflet area. And this was done by changes in the sort of the 4 marker positions. So you can see that with the power of 25 watts, we have almost a 30% decrease in the area of the leaflet. They did some histological analysis of these different power levels and showed that the amount of power that you deliver to the leaflet itself is proportional to the lesion that you actually observe in the end, which sort of makes sense. And what this would suggest is that you could potentially control or almost even titrate a reduction in size of the leaflet based on the application of the RFA.
Matt, can I ask a quick question?
Mm-hmm.
Looking at this histology, what kind of stain is this? And I'm a little surprised by this darkening.
Yeah, I'm pretty sure this is a Movat's pentachrome stain. And I think that it is, the darkening is sort of the fact that you're damaging or injuring or killing all the cells that are there. So it's this release of potentially cytoplasm that's happening.
Because traditionally black would be associated with elastin, right? In a pentachrome.
Yeah, I mean, I think it's more like that purpley black type color. I think it's due to the effects of the damage to the cells rather than you know, increasing in elastin content or anything like that.
Perfect. Appreciate it. Thank you.
Right, so they showed in a preliminary study here that RFA could be used to change the area of the leaflet. So then the idea then is, okay, if you have, you know, how could you actually deliver this in the body, right? You have a static radio frequency probe that needs to adhere for a certain amount of time to a potentially moving leaflet. And so a PhD student in the lab was sort of tasked with designing a way to actually deliver this, this energy to the moving leaflet. And what he developed was a sort of dual energy catheter. And the idea here is that he has built upon or sort of retrofit onto a standard cryoablation catheter an RF electrode. And what this would allow you to do is administer the liquid nitrogen into the cryoablation catheter, cause super cool temperatures that will stick the leaflet that's moving. And then once you have an attachment, you could then apply your radio frequency waves and then induce the shape change in the leaflet. He did some preliminary studies to show that you can have very stark gradients in the temperature with the attachment of this dual energy probe. So what I'm showing here, this is an image from his paper where he looked at a temperature image of the attachment of the probe. So here you can see this very, very cool -40°C where you're actually attaching. And then you have very high temperatures upwards of 80°C where you're actually administering the radio frequency ablation. Typically for tissue to actually start to change the shape and thermally to nature in response to temperature, you need to have an excess of around 50 to 60°C, depending on the literature that you cite. Here he was using a target value of about 65°C. He went on to show that you can actually change the shape of the valves ex vivo. So this is a system that he developed in the lab to put some markers onto a leaflet in a circulation flow loop and show that he could change the shape of the leaflet. But then also he did a preliminary sort of acute in vivo study where he performed these ablations in anesthetized animals and showed that he could get reductions of about 20 to 25% in some animals. Again, this depends on the efficacy of the actual attachment in which you're, which you have at the time. So this is sort of all of the preliminary work that's leading up to what I'm going to be talking about today. And here it really sort of sparks the motivation for our study. So previous work has shown that the RFA can modulate healthy tissue properties. And so what we wanted to know is, can this technology also be applied to a diseased mitral valve leaflet? Because ultimately that's what you're wanting to target is sort of the diseased patient population. The caveat here is that diseased myxomatous mitral valve tissue is often difficult to come by, and there are not many reliable animal models that develop this mitral valve degeneration naturally. Really, there's only one, which is a beagle, and it's sort of difficult to monitor, and it's difficult to actually identify, and of course, difficult to acquire those animals. So what we sought to do was to actually develop some sort of ex vivo approaches to develop a sort of quote unquote disease mitral valve. And so what we did is we took healthy porcine mitral valves and we sort of excised them in the standard way. And we performed a series of enzymatic degradations to actually see what would happen if we degrade collagen or elastin or some combination of the two in terms of the actual area of the leaflet. And so we used these concentrations of collagenase elastase, and again, this combination. And to give some representative examples here, so this would be contours of the leaflet over time. So sort of the light green would be an initial, and then the dark green would be sort of later stages in time. And so what you can see is that with an elastase treatment, the leaflet actually grows in size. We could then contrast that with what we found with our collagenase treatments. We saw little to no change actually in the overall area. And then when we combined collagenase and elastase, we saw a similar effect as what we saw with the elastase. We can, of course, measure these dynamics over time. What we see is that leaflets that were exposed to elastase, either in isolation or with collagenase, showed large increases in the normalized area of the actual leaflet itself. And if we compare this to a sort of range of disease mitral valves that we can find in the literature, this is consistent and could be at least an in vitro sort of approximation of an actual disease valve state. There are many caveats to considering this as a diseased valve, but we're sort of just saying that we're altering the structure in a way that increases the leaflet in a comparable manner.
What do you think are the mechanisms for that? Or are you going to go into that?
The mechanisms for the increase? For
the expansion.
I mean, the fact that we see the expansion with the elastase and the collagenase plus elastase, but not the collagenase, at a very surface level would suggest that this has to do with elastin degradation. I think that makes sense if you think about sort of this pre-stretch that exists inside the leaflet. And sort of as you release the pre-stretch from the elastin, that's sort of entrenching the undulation of the collagen fibers, you get this extension, and that will potentially lead to an increase. Okay, so as we, in our study, we used a combination of both collagenase and elastase as our sort of disease model. We also performed some mechanical analysis of these leaflets. So we can take the excised leaflet, place it into a biaxial mechanical testing system that is shown sort of schematically here. And then using our dual energy catheter, we can provide the RF energy. The thing here in our study is we didn't actually use the cryoablation, but we just sort of took advantage of the fact that this catheter had been retrofit to actually measure the temperatures that we're imposing. So we only used the radiofrequency ablation aspect here. So this is sort of a schematic of our system. And this would be a typical administration of RF power. We used a high value of power of 50 watts for 60 seconds. So we had this duration of on to off, and then we allowed for a sort of 2-minute relaxation time. And so you can see that as we administer this RF energy, the temperature will increase of the leaflet past this threshold to actually induce ablation. And that would look something like this. So this is a video of a sample that's actually being ablated. And so what you can see is that obviously the leaflet itself is shrinking down, but we actually have a pretty good tracking of these 4 markers as the ablation is occurring. And so we can see how the actual area based on these markers is changing. But due to the actual shrinkage in the different directions, we can measure the forces that this is actually generating. So this is our sort of mechanical analysis of the ablation itself.
Is the catheter on top of the tissue here or below?
It's on top of the tissue. So we're actually looking from underneath. Yeah. In terms of the mechanical analysis that we did, we used a sort of straightforward mechanical analysis of the leaflets. We applied a very small initial load to the leaflets and recorded an unloaded configuration where we marked the places of our 4 markers. We then applied 10 cycles of five different off-axis loading protocols to look at sort of a non-equal biaxial state within the leaflet. And those 5 loading conditions are shown here. We used a sort of tension as our threshold. So we used a ratio of 60 to 120 Newton per meter in the radial and circumferential directions. And then we had those different 5. And then after each of these 10 cycles for our loading protocols, we took the final cycle for constitutive modeling. The model that we used is a distributed fiber orientation constitutive model. I use a very generic term because some people may call this a Holzapfel model. Some people may call this the Gasser model. So it's a distributed fiber orientation model. It looks something like this. We have our invariant terms within this model. But the main thing I want to highlight is there are sort of 2 contributions in this model. The first of which is an isotropic contribution. It's typically thought to be due to elastin, sort of ground state matrix within the tissue. This could be the glycosaminoglycans as well. And then we have the second term, which is an anisotropic term. You can see from the exponential here that describes the behavior of collagen. And the behavior here is going to be dependent on our kappa term, this term kappa, where kappa is this distributed fiber distribution, or the fiber distribution of collagen. So after we performed our mechanical analysis and sort of fit our model to the data, we could then visualize what this energy function looks like. in a so-called stretch plot. So what I'm showing here is a radial stretch of the leaflet on the x-axis, circumferential stretch on the y-axis. And if we consider the dark portions here, this is the range of stretch in the radial and circumferential directions for a control leaflet before any RFA has been applied. You can see that we have about a 40% stretch in either direction. After the radio frequency ablation, you can see that these curves begin to shift outward, which would suggest an increase in compliance of the leaflet after the ablation has occurred. In particular, quite substantial increase in the circumferential direction, up to 1.8, and then in the radial even more. We could compare this with our digested leaflets. We can see again in the pre-RFA condition, we have a slight reduction in the sort of range of stretches, but similarly we see this increase after the ablation. Okay, so one other method that we used in the study was multiphoton microscopy. And so this is just an imaging modality that can be used to measure the intrinsic properties of the microstructural constituents in the leaflet. And so this is a schematic shown here of sort of a typical multiphoton system. The idea is that we have a laser that excites the sample, and then we have the ability to measure the sort of reflected light using different bandpass filters. And we can actually acquire two different signals from this type of tissue. The first is known as an SHG signal, which is intrinsic to collagen. This is known as second harmonic generation. And then the second is a multiphoton fluorescence signal, which is can be calibrated to the range of elastin. Elastin has a unique autofluorescence under visible light. So we can image these samples and get some sort of representative images that look like this. Of course, we were interested in microstructural changes associated with our digestion and the ablation. So what we wanted to then do was perform some fiber orientation analysis. So we did that using a sort of standard Fourier transform-based approach from which we could then recreate fiber distributions that would look maybe something like this in a polar visualization. The two main parameters that we were able to extract from these fiber distributions are shown here in red and black. So this value mu is sort of the main dominant angle of the fibers in the image. And then this kappa tells us something about the actual width of the distribution. I keep saying we did this analysis, but I actually used an open source software known as FiberFit. This was developed by the Lujan group at Boise State, which is a really good piece of software for people who may be interested in doing this. So I wanted to also sort of explain what our experimental design was here for this study. And really we wanted to look at two different effects. So the effect of the enzymatic degradation or the digestion on the actual properties of the leaflet, and then also the effect of radiofrequency ablation on the organization. And so our sort of experimental timeline or flowchart looks something like this. So we started with the isolation of the leaflet, and we performed an initial multiphoton microscopy assessment to look at sort of the baseline microstructural organization. We then performed our, so we randomly assigned control versus digested, and then we performed our enzymatic degradation, and then immediately performed a second multiphoton imaging. From there, we then mounted the samples into our biaxial testing system, performed a mechanical analysis, applied our radiofrequency ablation, collected the images that I had shown previously, performed a second biaxial testing analysis to show the effect of the radiofrequency ablation in the same leaflet, imaged again using our multiphoton, and then finally fixed and stored the samples. So they underwent quite a bit of testing each leaflet. But what this allowed us to do is to have really three specific comparisons of interest. So the first would be sort of from multiphoton to multiphoton here. And this would allow us to assess the effect of the digestion itself on the leaflet microstructure. So in terms of collagen and elastin. The second one would be to compare our third imaging session to our second. And this would allow us to see the effects of the RFAs, the ablation, and the digestion if we were to compare the gray and the green on the leaflet microstructure. And then the third effect would be by comparing our mechanical data. So we could see the effect of, again, RFA and digestion on leaflet mechanical properties.
Quick question, Matt. Did you unmount the tissue before you did the multiphoton imaging from the mechanical test device?
Yeah. So this is a limitation. So we did have to, remove them and remount them. We placed markers and things to be sure that we were in the same orientation every time, but there is that sort of effect of removal and replacement. Okay, so if we look at this first comparison, so this would be just the effect of the digestion on the microstructure. We can see that we have our control sample that looks something like this. We have a distribution of fiber angles. We have a dominant angle mu in our distribution kappa. This is actually also a fit to this distribution, the solid lines here. So we have our control sample. We can compare that to collagenase, and you can see that they looked relatively similar. This may be expected because we didn't really see any gross change in the actual overall area of the collagenase treatment. So we maybe wouldn't expect too much change in the microstructure itself. If you look at the collagen, you may be able to see some differences in sort of the size of the collagen fibers, which may suggest that there's been some degradation of the collagen itself. If we contrast that now with the elastase, we see that the fibers become straight. They lose their undulation or their waviness. And there's a pretty stark reorganization of the actual fibers themselves. So we go from a dominant angle of about 13 in this case to a dominant angle of about 46. So we have this reorientation towards the vertical axis in this image, which is the radial direction of the leaflet. Similarly, if we compare our collagenase and elastase group, it looks very similar to our elastase group. Again, that's consistent with what we saw in terms of the overall area change. If we then wanted to look at now the effect of the ablation itself on the leaflet microstructure, we can acquire our same images, and we can see that if we have our collagen fibers before the ablation and our collagen fibers after the ablation, we see some similarities. We see that we lose this undulation, the fibers become straightened, and then there's sort of this radial reorientation of the fibers themselves. We wanted to look to see how large the effect of the ablation actually was within the leaflet itself. And so we always performed the ablation in the center of the leaflet on our biaxial testing device. But when we were imaging, we have the ability to image at multiple locations. And so for a subset of our samples, we looked at 5 different locations around the leaflet itself. And we saw very similar things in all 5 of these regions. We see an accumulation of the fibers, so they become the distribution becomes narrower, they become more aligned, and the angle always tends to reorient towards the radial direction in this case. We can, of course, measure these changes in the fiber dispersion in the dominant angle, and we can see that while the dispersion maybe is less sensitive in terms of statistical significance, the dominant angle is very clearly increasing after ablation in all 5 of these different regions. We can then also look at the effects of collagen orientation after both the ablation, but also after the digestion itself. So what I'm showing here are sort of 2 effects in total. So we have our pre-RFA group, the control in the dark gray and the digested in the light gray, and our post-RFA group, again, control and digested. We can see that for the fiber orientation, the greatest effect here is due to the actual ablation itself, rather than the digestion in this case. The digested samples seem to reorient a little bit less on average than the control samples. And you can see that here. So this is a sort of paired measurement in the same leaflet throughout those different portions of our experimental design. And you can see that, maybe on average, the control samples tend to reorient a little bit more. They have a larger change in the angle.
What is the physiological relevance of this dominant angle?
Physiological relevance, that's a good question. You may think of whenever we were doing the study, we were thinking of, the ability to actually deform in different directions. So if you have potentially a reorganization towards the radial direction of these fibers, there may be, less stretch in the radial direction, and that may have some effect on coaptation, for example. But that's a really good question. I mean, that's always the question in these, fiber-based models. What is the relevance of the measurements you're making?
But you can't control it, right? That just happens to be one parameter of your fiber distributions that significantly were affected, yeah?
Right, yeah. That was another thing that I'm gonna say at the end, is that we didn't really try to manipulate this at all. We were just applying the ablation and we're observing what's happening.
Thank you, Matt.
Mm-hmm. If we look at the dispersion, we can see that if we look in our pre-RFA group, the dispersion is already affected by the actual enzymatic degradation itself. So we have this the fibers become more oriented before any effect of the ablation actually occurs. And we can see that in the digested groups, they remain essentially the same. So very little change in the actual dispersion of the fibers themselves. Again, it's a good point that was brought up about the actual relevance of what these mean, but we were just sort of observing what the effects of digestion and RFA have on the actual microstructure of the leaflets themselves. And what we can say here is that there's a clear alteration of the organization of both control and the digested microvalve leaflets. So I mentioned also that we can measure the dynamics of the ablation in terms of the force and the area change that's associated with that. In our biaxial testing device, that may look something like this. So we have the change in the radial length in blue, the circumferential length in light gray, and then the overall area in black for our control leaflets. And associated with this ablation, you can have our sort of increase in force during the ablation and then during that relaxation period. The ablation is between these dashed lines. So you can see that as the leaflet is shrinking, the force is accumulating in both the circumferential and the radial directions. And that drops back down after the ablation is stopped. In the digested samples, we see similar trends in terms of the area change. One of the most stark changes that we see is the amount of force that's generated in the radial direction after the ablation. We can look at the overall change in the area. It turns out that the digested valves actually reduce their area a little bit more than control. This could be because they're so much larger to begin with. And also the rate of change of the area, so that this sort of initial slope is a little bit larger, significantly larger in the digested group, which may suggest some sort of susceptibility to the actual ablation itself. Again, the radial force is significantly increased, whereas the circumferential force is not statistically different. We think this probably has to do with, again, this radial reorientation of fibers. And as you begin to denature the fibers, you're actually pulling more in the radial direction in the digested samples. We also performed, again, our biomechanical analysis. We can look at sort of standard biaxial stress curves for their samples, both before and after the ablation. The main thing I want to highlight here is the change in the circumferential direction here. So what you see is before ablation, this dashed line, and after the ablation, we have this rightward shift of the stress. In the digested samples, this is much more apparent. So we have this increased compliance in the circumferential direction after the ablation. We can look at differences in the actual stretch themselves. And so what we did was we took a fixed stress value of 100 kPa and then pulled the stress value or stretch values at each of these for each of these curves. What we see is that there's little effect in the radial direction. There's actually this increase in the digested samples, which is a bit interesting. And what we see is more pronounced change in the circumferential stretches. From here, there's a couple of other metrics that we pulled out, one of which is known as the stored energy. So this can be thought of as the area underneath of the curve. And this stored energy, as you imagine, as this curve moves to the right, the area underneath of that curve is increased. So all of our ablated samples tended to store more energy. We also looked at the stiffness, which is sort of a linear contribution, again, at this working point. What we saw is that with the ablation, the stiffness was dramatically reduced in both the control and the digested samples. Finally, we wanted to look at sort of the quote unquote therapeutic potential of this in our ex vivo system. So what we did was we, again, performed our analysis in the control and digested samples, but this time we took the contours both at the start of the ablation and at the end of the ablation. And what we found was that the main finding here is that the area is restored back down to the levels of the control. So if we look after the ablation in our control samples, we see that the area is decreased. We can see that pretty evidently here. The digestion itself, we know that that's going to increase the area. That is shown to be the case. But after the ablation, these digested samples are returned back down to the area approximately of control, albeit the shape is a bit different. One thing that is maybe not a great thing is that the thickness of these leaflets is increased and maintained. So this is maybe not ideal. But if we combine the effects of the area and the thickness, we can see that overall the volume of our treated leaflets are restored again back down to the levels of control. So this would maybe suggest that there's a sort of therapeutic potential of this ablation. But there's much more to be done here in terms of understanding the actual clinical use. Just really quickly for the sake of time. So key findings, the digestion of elastin, we think, tends to promote the leaflet enlargement ex vivo. The digestion alters the collagen fibers and results in this radial alignment. Ablation induces reorientation throughout the entire leaflet itself. And the digested leaflets show a large increase in area and greater radial forces during ablation. We think this has to do, again, with this radial reorientation of fibers. We have an increase in the circumferential compliance, and that leads to an increase in energy storage, which is an interesting mechanical finding. Again, the clinical relevance maybe we need to understand some. And overall, we see that the ablation is able to reduce the area and volume of these digested leaflets. So there are a couple of future directions and questions moving forward. One of which is this precision ablation. We showed earlier that you can actually titrate the amount that the area will change with different amounts of power. And here we just applied a very large amount of power. So there may be some ability to actually control how much the area of the leaflet is changing and where. One thing that, again, is unknown is what is the long-term impact of this lesion formation that you're actually inducing in the leaflet if you're essentially killing off all the cells, to have this shape change. We didn't consider at all the actual thermal, like modeling the thermal properties of the collagen here, which may be an interesting thing to consider in the future. And then, of course, the biggest question in here is how does our in vitro model actually compare to the mixed homogeneous leaflets that are developed naturally? Okay, so with that, I will stop. Again, Josh and Wilson are really the main ones who did this. And I'm happy to chat with anyone about science anytime. So this is my email down at the bottom. Thanks.
Thank you, Matt, for this overview. Clearly a beautifully designed experimental study, and you seem to have a lot of data to go down many different paths. Who has a question? We already had some, but I'm sure there are more. Feel free to just unmute yourself and go straight ahead.
But if you're shy, you can also just type it in the comment section.
Yeah. Ignore my laundry behind me. Hey, Matt, how are you doing? Good to see you.
Good, how are you? Good.
Good. Hey, quick question regarding your multiphoton microscopy. Are those all images reported from a single depth within the tissue? Is that like atrialis? And
yeah,
if so, did you see differential effects within the fibrosa? Like, did the fibrosa have that radial alignment too?
Yeah, that's a good question. So the images that I reported were always from a standard depth. And we were just trying to get the most uniform depth to present the images. Whenever I was reporting the distributions, for example, those are averaged throughout the entire thickness that we're able to get. We did not do any sort of clearing or any sort of alterations that way to get, further down penetration into the tissue. So we were about, 50 to 100 microns in depth on average.
Thank you very much.
I guess I have a question. First and foremost, thank you for that great talk. I thought it was really interesting. I have a handful of questions, but I guess I'll ask them in the order that I think are the most important. Is there any concern over the denaturing of the interstitial cells? Because it seems like you're blasting these valves, and I don't know if these cells can survive. And I guess, like, in the context of the surgery, I imagine you probably don't care about them because the people who need this surgery are probably pretty old, right? I just wanted your opinion on that.
Yeah, that's a great point, and I think Manuel also was leaning toward this. So here, I've shown several studies, but really this is still proof of concept, right? We've never really done a long-term in a patient study. We did do some sort of percutaneous approaches in pigs, but they were all acute studies. And so I think that, yeah, the cellular biological effects here are really, really not known. And what are the long-term effects of that? Who knows? It could very well be that when you're ablating these cells, right, you're sort of making this a more thrombogenic surface, right? So that there may actually be more long-term complications, but those are things that could potentially be ironed out with further studies. Here, we were just trying to show the efficacy of actually, can you reduce the size of a valve in a way that doesn't leave something in the patient?
And I guess like a follow-up question to that, do you predict that RFA treatment can potentially it might lead to just a different pathological state? Like not myxomatous mitral valve, but not the native tissue and just like a different disease?
It could, I mean, it could, it could. Again, I think without the lack, with the lack of like more biological mechanistic studies, it's hard to speculate about what would actually be occurring there. There are certainly ways to sort of culture leaflets, excise them and culture them ex vivo. So we could maybe apply the actual ablation, culture them for, again, maybe a short amount of time and see if there are any cells there at all. Do the cells recover, that are from the periphery, things like that. In adulthood, things like regeneration of soft tissues don't occur all that much. But, even now there are some potential approaches to like reprogram cells in vivo to actually induce more of like, not necessarily a stem cell-like state, but a more regenerative state. So there could be it could very well be that the ablation is not good long-term biologically, but there may be some other sort of co-treatments or something down the line that would be that would improve it further.
Matt, I have a
Alex, do you have-
philosophical. Oh, sorry.
No. I was wondering if Alex had more questions.
I do have one more.
Oh,
Do you predict that in the future, the spatial control of RFA treatment will be such that we can actually morph the leaflets back to their original shape? Because it seems like what you have now, you can reduce the area, but it looks like spatial control is limited. And I know you only tested like one really high energy radio ablation, but I just was wondering what your thoughts were on that.
Not being an interventional cardiologist, I would say, yes, totally. You could probably do that with all of the totallys and probablys that I can add. In terms of like control of catheters, the technology is really, really advanced. I mean, the MitraClip approach, there's a huge number of degrees of freedom. So you can certainly have precision in how you're positioning the catheters. There's no doubt about that. That's sort of one thing that I mentioned at the end there is that like we gave the high amount of power, but previous studies have shown that if you give less power, you can actually adjust the shape less. And so there's certainly a way for, a way forward that would be more precise. I would certainly think that could be possible.
Good, thank you. I have one last philosophical question, and hope I don't steal Manuel's thunder. But it seems to me that a lot of surgical approaches, especially to treat valve disease, what it does is at times it kind of just pushes the problem away. It doesn't address the underlying issues. And I think the underlying issues is like pathological behavior of the interstitial cells, right? And I think that might be the end all be all in terms of solving a bunch of valvular heart diseases. And like you were saying earlier, it could become in the form of some sort of gene therapy or something. But what is your opinion on the current state of like surgical treatments to treat heart valve disease?
Yeah, I think I said it in the actual presentation. I mean, like there's no targeted therapies for valve disease. And that's sort of what you're alluding to, it's this pathogenic state of the cells that are there. So that there's, I think people understand, a few pathways that may be regulating some of that, maybe at a surface level. But to talk about like precision medicine and patient-specific treatments, I think we're frankly still a ways away for doing anything with valves. That said, the surgical approaches do work and they're successful now. And typically the patients are older and typically they have other comorbidities. And it's like you said, you're kind of kicking this thing down the line. It's a similar concept with like a joint replacement or something like that. There's really only efficacy for like 15, 20 years. And so for an older patient population, do you really need more than 15 to 20 years? So I think there's a lot that can be done and a lot to do further, but a lot of the surgical approaches now work and they work well. So I don't know if that answers your question.
Yeah, thanks.
I would actually have a question. ablation, the way that I know I was most familiar with, is used in the heart to change the electrophysiology of the heart. Are you aware of people analyzing how the muscle cells or the tissue changed in terms of geometry following ablation? Because it was not necessarily the primary focus, because it was more the electrophysiology. Do you have any comparative studies there that you know of?
So when we were writing the paper, I did look for those studies to see, is there something outside of this, frankly, small group of studies that were all done by the same people? Is there more out there in the literature? And I really couldn't find much. It's more like when you look in the electrophysiology literature, it's more like the shapes and the patterns that they actually do for certain types of fibrillation. It's very clinically focused. It's not really more focused on, what are some of those tissue level changes per se.
All right, thank you. Manuel,
And
your
I have a little bit of a philosophical question for you.
Okay.
So Adrian and I have recently done very similar work applied to skin, and not with radio frequency, but just sort of hot water treatment. But one of the things that I think and I think Adrian as well struggle a little bit with is that as you denature the collagen, right, you produce forces. As you not digest but denature through the heat application, you produce forces, right? And so we've been trying to think about what is the mechanical basis for this, in that you model your tissue as hyperelastic, right? And you assume that the forces produced are sort of elastic forces. But when you really think about collagen being denatured and really undergoing a configurational change, one of the questions we had is, would it be more accurate to really look at this as an entropic spring? In other words, as your tissue loses organization and entropy increases, but say you keep the boundaries fixed and tractions applied, is it really you preventing it undergoing a energetically more favorable configuration that produces the forces? Does that make sense? And have you thought about that sort of conflict with our energy-based modeling approach rather than looking at it from an entropic perspective?
Yeah, so it's very interesting. And if I were to approach something like that, I think that the thinking that you and Adrian have is probably at least the first approach of the right way to go. That said, I haven't really thought about how to model this or any of that on my own. I was more focused on more standard approaches, at least initially. I will say that along those lines, in terms of the entropy and maybe what is actually happening to like a collagen fiber or even at the fibril level, combining some of this with imaging like spectroscopy, like Raman spectroscopy, things like that, you can actually look at primary, secondary, and tertiary structures of a given collagen fibril itself. And so there may be some insight there. I know that there are studies where they've given collagenase an image under Raman and shown that some of these structures actually change, right? And you can detect that at sort of that spectroscopic level. So yeah, I'm not sure exactly how to approach that, and I'm not thinking about that myself in terms of modeling this, but that could be a direction to start.
Thank you.
Hello. I've got 5 million questions, but I'll probably only ask 1 or 2 depending on time constraints.
Okay.
So the first one, and you may have addressed this, forgive my confusion if so. I know that y'all's main intent was to reduce surface area of the valve because it was too large and not closing properly. But when you do that, wow, I lost my train of thought. I'll skip to the next one. Okay, so cool. So I noticed that when you had the slide where it showed the fiber orientation, it had what looked to be pretty aligned like fibers after the RFA, but the compliance actually showed the exact opposite, where it was highly compliant compared to the normal. which those two kind of facts seem counterintuitive. I know that there was degradation of some tissue, which would reduce some stiffness, but why would that be the case?
Yeah, so that's a good point. And I think that kind of comes down to the fact that there was a lot of information and I was going kind of quick because I was looking at the clock. But so you're right. So the fibers become less undulated, they become straightened, they reorient to the radial direction. So that was like vertical in the image. The compliance that I was talking about was in the circumferential direction, so perpendicular to that.
Gotcha.
So you can imagine that if you have straight, stiff fibers and they orient vertically and you're pulling horizontally, the compliance would maybe increase.
Yep.
So that's what we suspect is happening.
Okay. So I remembered my other thing. So if you reduce the area, but you increase the compliance, since this is going to be under force in the body, wouldn't that basically have the same effect potentially when under stress, like stretch much more to where you have the exact same problem?
Again, it could be, and I think that this is where more long-term applications of this are going to be crucial. But again, I mean, when you're talking about coaptation of the leaflets, that's primarily going to be occurring in the radial direction, primarily.
Okay.
And so the compliance that we saw was again in that sort of circumferential direction.
Okay.
If there, you can imagine all sorts of scenarios where if you don't apply the ablation properly, maybe the coaptation would not be ideal, or maybe the regurgitation would not be eliminated, but only reduced. So yeah, those very insightful questions. I think it just requires more experience to really answer.
Okay. Thanks.
All right. Is there another pressing question? Otherwise, we're at the time. Yeah, Caleb, go ahead.
Hey, Matt, have you? I know this is outside the realm of your study, but thinking of doing this in situ where you have this blood flow coming around, is that does that alter the heat that you're delivering through the RFA, or does the localized energy delivery, will that alter the properties of the blood itself, or do you think the blood flow is so rapid that it's just going to dissipate and not have a huge effect?
Yeah. So the blood flow certainly does have an effect. So if you don't have proper attachment of the actual ablation probe to the tissue, the energy just is delivered everywhere. And what actually happens if you do this in a stream of active blood, it actually coagulates the blood. And it doesn't coagulate to the extent that there's, you're blocking everything, but it coagulates on the tip of the probe. And so it impedes actual delivery of the energy. In terms of like the effect of the blood flow and maybe diffusion of heat or something like that, I don't know that would have too much of an effect. The amount of heat that you're giving is so high relative to the body temperature that if you have proper attachment, there's really no reason to expect that the temperature decreased due to the blood flow.
Thanks.
All right, Matt, thank you so much again for this amazing talk. Cool discussion afterwards. And thanks everyone for joining.
Thanks, Matt.
Mm-hmm.